Okay, so what we're going to look at today is gene probes and medical diagnostics. And basically just a quick look at what gene probes are, why we use gene probes, and the different ways that they can work. So essentially, the whole idea about a gene probe is for us to be able to look for alleles. So a DNA probe and hybridization is one way that we can look for alleles. And what DNA probes can do is they can locate a specific allele. They can locate specific alleles of genes or see if a person's DNA contains a mutated allele that causes a genetic disorder. So what we're going to quickly look at first is what actually a DNA probe is. So we've got a bit of an example here, and we'll go through this in a bit more detail in a moment. So what is actually a DNA probe? You'll sometimes see DNA probe, gene probe being used interchangeably, and that is fine. So a DNA probe. What DNA probes are is they are a short strand of DNA. So they're really, really tiny, short strand of DNA. And you can see that on the image on the right here. We've got this really small, short strand of DNA here. And this short strand of DNA has a specific base sequence. And this specific base sequence is really important because this specific base sequence that it has is complementary to the base sequence of the target allele that we're looking at. So it's got a specific base sequence, which is complementary, and that's really important, specific base sequence, which is complementary, so I'm just going to write comp, to the target alleles base sequence. That's our DNA probe there, and it has a specific base sequence that is complementary to the target alleles base sequence as well. And basically, how it works, well, what this target allele might be, is it might be the allele we're looking for might be one that causes a genetic disorder, for example. And what happens is this DNA probe will hybridize, and by hybridize we mean binding. So the DNA probe will hybridize to the target allele if it's present. That's really important. That's if it's present. So it's not going to hybridize all the time. So if it's present in the sample DNA. So if that sample DNA that we combine with the gene probe, DNA probe has that target allele, the gene probe, the DNA probe will hybridize to the target allele if it's present. And then the last thing that we then need to be able to do is actually be able to visualize this, because it's great this DNA probe actually attaching to it. We've got this bit of extra DNA, but we need to actually know that it's there and how it works. So what we then need to do is we need to be able to visualize this. And a DNA probe has a label attached to it, and it will be one of two types of labels. And this label allows us to visualize if this hybridization has occurred. The two types of labels that could be added are, well, there's more than two, but the two most common that you need to know about is one would be a radioactive label. And that would be found using X-ray film or fluorescence. Detected using UV light. So what we're going to have a quick look at is how we actually attach these labels and how it actually works and what actually happens. So how we actually attach these labels to these DNA probes. I'm just going to use this image here. So this is an example here of a fluorescence that's had DNA probes. So gene probes have been added to these samples, and where they are fluorescing is where those required alleles are. So that's where that hybridization has occurred. So when we attach these labels, the first thing that has to be done is the DNA has to be digested by restriction enzymes. When that DNA is digested, we use restriction enzymes, and then we separating using gel electrophoresis. So the DNA is digested, and that's using restriction enzymes. And then, like we've done lots of times before, we then separate it using gel electrophoresis. I'm not going to go through gel electrophoresis. We've done that a couple of times in some of the other videos. So if you're not sure, have a look back at a couple of the more recent recombinant technology videos and making DNA samples and fragments videos, and it'll be in those. So our DNA is digested. It's digested using restriction enzymes, and then it is separated using gel electrophoresis. So we have all those parts of the DNA separated out. These separated DNA fragments, these are then going to be transferred to a nylon membrane and incubated. So we're going to go through that southern blotting process. So we transfer to a nylon membrane. We incubate this nylon membrane, and then we add the fluorescence labels. It's incubated with the fluorescence labels. So we transfer to the nylon membrane and incubate with the fluorescent labels. Then, as we've already said, if the allele is then present, it will hybridize and bind to it. So this is exactly the same as we said before. If the allele is present, we'll have a hybridization occurring. It will hybridize. If we then expose that membrane, like we can see here on the image, to UV light, if the gene is present, there'll be a fluorescent band. So there'll be fluorescent bands under UV light. So for example, on this image here, you've got your marker ladders here, which have obviously all got the DNA tags on. And then we've got different, I've literally just found one offline. It doesn't really matter what it is that they're looking at, but where they've tagged different viruses, it looks like, with a fluorescent DNA probe. The reason I did this was just to show you how it looks, so you can see the different areas of fluorescence coming up. So you can see those bright bandings here where you can see the fluorescence, once it's shown under UV light, appears. So that is one way we can look for alleles using DNA probes and hybridization. You could do exactly the same thing and just use it with X-ray film as well. So rather than it looking like that, it would look just in those black and white versions, very similar to the DNA fingerprinting. But like we said with the DNA fingerprinting, we can use fluorescence or radiation, so X-ray film, same as this. Alternatively, the probe can be used as part of something called a DNA microarray. And microRNA actually screens for a lot of genes at the same time, and that's what we're going to look at now, once I can find my microarray. So this here is a microarray. I'm just going to spell this at the top for you. So microarray. Okay, so DNA microarray is able to screen for a lot of genes at exactly the same time, which is really, really good, really useful. And basically what this microarray is, as you can see it here on the screen, it's just a glass slide. And this glass slide has microscopic spots of different DNA probes attached to it in rows. So these DNA probes are already attached to this microarray. So it's a glass slide with these little tiny wells in that are attached. When you see one in real life, they're super, super, super tiny. And we always make them look bigger on pictures so we can explain it easier. So yeah, it's a glass slide, microscopic spots of different DNA probes attached in rows. So we've got different probes in this one. So I'm going to just mark that down because that's quite important. So we've got different probes in the rows. So what we would then do is a sample of fluorescently labeled human DNA is washed over the array, and we wash it over the array. So we wash, in this example, we're going to wash a sample of fluorescently labeled human DNA sample of, I'm going to put F labeled human DNA. So we wash it with a sample of fluorescently labeled human DNA, which will stick to the array. What we then do is the array is then visualized, and as you can imagine, it's visualized under UV light because it's fluorescently labeled. So we visualize it under UV light. And as you can tell from the time, exactly the same as when we were using just the individual DNAs with just one probe, the labeled DNA attached to a probe will show up. It will fluoresce. Any spots that you see on a microarray, and I'm going to actually talk about this image now, means that the person's DNA contains that specific allele. So you will, if we're using lots of different DNA probes, we will label them with different fluorescent colors. So as you can see on this one, we've got 3 different colors. We've got gray, which is just not present. There's no fluorescence going. Yellow, which in this example is present in both cells. Green, normal cells only. Red, in disease cells only. So you've got 3 different fluorescence going on, basically. So in this one, as you can tell, they were looking at a few different things here. So they were looking at normal cells, and normal cells, they tagged with green fluorescent probes, and then cancer cells, they tagged with red fluorescent probes. Well, red fluorescent probes were looking for cancer cells, green fluorescent probes were looking for normal cells. Mix them all into the microarray. And then this is what you got. Obviously, red and green together make yellow. So what happened was, as you can see here, these cells here, there was nothing present. So they weren't significant. Obviously, hybridization hadn't occurred, nothing present. Then we were finding that these ones were appearing only in normal cells, these ones in disease cells only, and then these ones in both. So that is how a microarray would look. You get basically a square with lots of circles on in different colours, and then you have to understand what those different colours mean to understand what's happening. Great thing about microarrays is that we can look at more than one, we can attach more than one probe at a time, so we can look for more than one allele at a time. To produce a DNA probe, you need to sequence the allele that you want to screen for. So like when we were sequencing before, if you can't remember how we do that sequencing, you need to go back and watch the making DNA fragments and genome video. And then you use PCR to produce multiple complementary parts of the allele, and these are then the probes. So what are some of the uses of these DNA probes? And this is going to be a quick overview of why we might use these DNA probes. So why do we use them? So let's have a look at the uses. What are the uses? Right, so screening. Screening with these DNA probes can be really, really useful. We can use it for looking for inherited diseases, inherited conditions, inherited diseases. So, for example, Huntington's disease could be used using these microarrays and using these DNA probes. So Huntington's disease is an inherited condition that affects the nervous system, and it doesn't usually start to display symptoms until a person's age between 30 and 50. People with a family history of the disease may actually be screened for the mutated allele. So we're looking for the mutated allele for Huntington's disease because it's got a mutated allele. We can look for that in someone's DNA to find out if they've inherited it. The NHS also offers to screen all new babies for inherited disorder, cystic fibrosis. So we could also look for cystic fibrosis. And cystic fibrosis, as we've covered before, can cause breathing and digestive difficulties. So treatment of that condition can begin as soon as possible. Another thing that we can use these DNA probes for is to determine drug response, and it's that design a drug idea. So, for example, the idea of these determining drug responses, see how a patient will respond to specific drugs in their body. So, for example, if we talked about breast cancer, breast cancer, as we know, can be caused by a mutation in the HER2 proto-oncogene. And this can be treated with a drug called Herceptin. Make sure I write that nice and clearly. Treated with a drug called Herceptin. However, Herceptin is only effective against this HER2 mutation. We know there's lots of other mutations that can cause breast cancer, like BRCA1, BRCA2. Herceptin will not work against any of them. So being able to run someone who's got breast cancer, their DNA, using one of these DNA probes, a microarray, to see if they have the HER2 mutations, great, because then that lets us know if there's any point us treating with Herceptin or not. Otherwise, and we might be wasting time, money, and making someone more poorly because we're treating them with a drug that isn't going to work on them. Third reason and use of these DNA probes is health risks. And what we mean by health risks is actually looking at how likely someone is to inherit a mutated allele. And that might be during genetic counselling, if couples are wanting children, etc. So inheriting a particular mutated allele increases your risk of developing a certain type of disease. So, for example, if a person knows they have these alleles, it might help them make a choice that could reduce their risk of developing this disease. So there's two ways we can look at this. We talked about like that pre-implantation genetic diagnostics. I'm going to look at it this time as actually an adult screening themselves for their own health risks if they're going to develop a disease. So certain types of cancers, one type of cancer that could be, that we know can be inherited, is the BRCA1 gene. If somebody knows they've got the BRCA1 gene, they have a higher increased risk of developing breast cancer, substantially higher than anybody else in the population who doesn't have the BRCA1 gene. So the genetic screening will then allow these people to make those decisions of actually, well, some women and some men who have the BRCA1 gene might decide to then go and have their breasts removed. So these preventative measures to ensure that they then don't develop breast cancer further down the line, it's preventative rather than curing, because we're stopping it before it can happen. There is some concerns about this when we're using DNA probes for health risks, because some people worry that genetic screening might lead to discrimination. So I'm going to just pop here concerns over discrimination. That's by things like insurance companies or employers, because if they know you've got a high risk of developing a condition, they may put things in place maybe to stop you working for them or charge you more money, for example, if they were an insurance company. So the last little bit that we're going to look at before we go through some exam questions, there were the three main uses: inherited conditions, determining drug responses, and health risks, is another quick look at genetic counselling. I'm going to just give you some examples. In the exams, you will often be asked to evaluate information about screening for inherited conditions and people's responses to drugs. So you need fors and againsts. So it's really important to think of those ethical concerns around these. genetic counselling, as we've covered before, and we'll do this really quick as a recap. Genetic counselling, it advises patients and relatives about the risks of genetic disorders. So it's just all about risks of genetic disorders. And what genetic counselling does, it gives advice about screening and then results explanation. So a genetic counsellor is someone who, before you go and have any genetic testing, explains the genetic tests, explain the risks, explain what you might find. Then, once you've had the genetic tests, they explain the results to you. They then explain your options. And the whole idea around genetic counselling is that this advice can be given, if positive, to someone on a preventative focus rather than on a treatment focus, or if they are already suffering on treatment. So the idea of genetic counselling, if it's positive, we can then look at prevention or treatment for people. So I'm going to just talk through an example. So a woman with a family history of breast cancer may go and have genetic counselling to help her decide whether or not to be screened for known mutations that can lead to breast cancer. So like that BRCA1 gene I was just talking about. If she's screened and the result is positive, the genetic counsellors might explain that a woman with mutated BRCA1 gene has a 50 to 85% chance of developing breast cancer in her lifetime. And counselling could also help the woman to decide if, for example, she wants to take surgical steps to reduce the risk. Another example is sickle cell anemia. Sickle cell anemia is a recessive genetic disorder, and that's a mutation in the hemoglobin gene. A couple who are both carriers of the sickle cell allele may like to have children. They themselves don't have sickle cell anemia, but they may undergo genetic counselling to help them understand their chances of having a child with sickle cell anemia. Their chances are 1 in 4. Genetic counselling also provides unbiased advice on the possibility of having IVF and then screening their embryos. So that's then offered as an option. There's again lots of ethical issues around that, but that is an option for patients, and they give them all this information. It could be, well, you might choose not to have children. You might choose to have children, just risk them having sickle cell anemia. You might choose to actually go down the IVF route and we screen the embryos for sickle cell anemia. And so if the embryos have sickle cell anemia, they then would be implanted into the womb. And it can also provide information on help and drugs available if the child was born with sickle cell anemia. What we're going to do now is just look at a couple of exam questions based around this whole topic of genetic technologies and DNA probes. What we're going to do now is look at some exam questions, have a talk through them, pick out the key points that we would look at in the question, and then go through some model answers. So this first question is talking about scientists who wanted to measure how much mRNA was transcribed from allele A of a gene in a sample of cells, and this gene exists in 2 forms, A and A. So straight away in this question, I would highlight the word mRNA because that's a key point. We're not looking at DNA, we're looking at mRNA, first bit of information. And we're looking at how much mRNA was transcribed from allele A of a gene in a sample of cells, and the gene exists in 2 forms. The scientists also isolated mRNA from the cells, and they added an enzyme to mRNA to produce cDNA, and that's really important. So I'm just going to change the color of this to highlight that, to then produce cDNA. The first part of this question asks us to name the type of enzyme used to produce the cDNA. So when we think back to what we've already talked about in this topic, when we're using cDNA, if we're making cDNA, which is that complementary DNA, the enzyme that we use to produce cDNA is reverse transcriptase. And that is something that should come straight to the top of your head. Okay, we're isolating our mRNA. We're going to make cDNA. What are we going to need? Well, the enzyme that we're going to use for that is going to be reverse transcriptase, and that would get you 1 mark. It's a nice, quick recall question. Also gives you the idea of what the rest of the question might be about. The rest of the question then goes on to then start to just give you some more information of this experiment. So the scientists used a polymerase chain reaction, PCR, to produce copies of the cDNA. They added a DNA probe for allele A to the cDNA copies. This DNA probe had a dye attached to it, and this dye glows when a green light only when the DNA probe is attached to its target cDNA. Explain why this DNA probe will only detect allele A. So explain, we need to give, it's a 2-mark question, so we need to be given 2 points. So we're needing to say something about the probe and then something about allele A. So explain why this DNA probe will only detect allele A. And that only is actually really, really important. They used PCR, so that's quite good to keep in mind. To produce copies of the cDNA, they added a DNA probe for allele A. The DNA probe had a dye attached to it, and the dye glows with a green light only when the DNA probe is attached to its target cDNA. So why does this happen? Well, we've just talked about DNA probes. So as we know about probes, a probe's base sequence is complementary to that of the DNA that we're looking for, so the allele that we're looking for. So our probe's base sequence is complementary to the DNA of allele A. You need to make sure you're saying of allele A to make sure that we're linking it to that. So the probe's base sequence is complementary to the DNA of allele A, and it binds together, as we know, because they're complementary, they'll bind together by forming hydrogen bonds. So they have that binding together, forming hydrogen bonds. That will get you the first part of the mark. Then, so explain why the DNA probe will only detect allele A. Well, we've explained what a DNA probe is basically in this first part question. We're saying a DNA probe has a base sequence which is complementary to the DNA of allele A and binds by forming hydrogen. I've missed the word bonds out. Hydrogen bonds. That's your first mark. Your second mark is then going to explain this word only. Why is it only detecting allele A then? And that is, so because it has a base sequence which is complementary to the DNA of allele A, it binds by forming hydrogen bonds. So only this DNA, and by this DNA we're referring to the allele A DNA, is labeled. So it is the only one to give off the green light. And that is your explanation point there. So what a probe is, got that complementary base sequence, they bind together. So because they do this, that probe is going to only be attached to that DNA, so it's only going to be that DNA that's going to fluoresce, or in this case, give off a green light. So that is the second part of that question. Then goes on to show us some results. So the scientists use this method with cells from 2 people. 2 people are named, were given the initials H and G. One of these people was homozygous dominant, so let's highlight that, and the other was heterozygous. The scientists used the PCR and the DNA probe specific for allele A, so the dominant allele, on the cDNA from both people. And the figure below shows the scientists' results. So we've got our intensity of green light over here, our number of PCR cycles, using cells from person H and using cells from person G. So it's looking at how much light is given off by each individual. The question then asks us to explain the curve for person H. And I would highlight that person H to make sure you're answering about the right person. So what's happening? Well, the curve for person H is increasing. So why is it increasing? Well, the more probe binding that's happening, the more the cDNA is there, there's more allele. Gene A means there's more light. So explain the curve for person H. Person H has more probe binding. That will get you 1 mark. You've described what's happening. They've clearly got more probe binding. And we know that because, oops, let's write with a sensible size pen. Because more gene A equals more light. What is happening to this? Well, actually, as we look, our DNA with A doubles each PCR cycle. Our DNA with the dominant allele doubles each PCR cycle. So that will get you another mark. And then finally, what I would say, I'm just going to write it up here. So the light will approximately double each time. So the curve's going to steepen more and more. The curve's going to go up exponentially. It increases even faster. Something along those lines to get you that 3rd mark of saying, so the light levels will approximately double. Remember person, and that was person H. So then you're asked, which person, H or G, was heterozygous? Explain your answer. Well, the light given off is only given off by that dominant allele. They're obviously both going to be given off light because they both do have that dominant allele. Person H has got more light being given off than person G. So you would assume that person H is going to be homozygous. And so then person G is going to be heterozygous. So first part of this question, who is heterozygous? 1st mark, even if you don't put anything else, just put G, you will get 1 mark. Now you are asked to explain your answer to then get your 2nd mark. So G, and that's because heterozygous, so heterozygous only has half the amount. And it has half the amount of the probe for the dominant allele. Probe for A. So will only produce half the amount of light. I'll go for it again. 1st mark, G is heterozygous, so only has half the amount of the probe for A. 2nd mark, so will only produce half the amount of light. So that's how we know G is AA, heterozygous, and H was homozygous. We're going to look at question 3 now. In this question, a husband and wife wanted to know whether they were carriers of a mutated form of a gene. This mutation is a deletion, so that's important to note. And it causes a serious inherited genetic disorder in people who are homozygous. Geneticist took some samples of DNA from the husband and the wife, and he used a DNA probe to look for the deletion mutation. The DNA probe was specific to a particular base sequence in the exon in the gene and exons of coding sequences in a gene. The geneticist compared the couple's DNA with that of the person known not to carry this mutation, and the chart below shows the geneticist's results. So amount of DNA probe binding to DNA sample. So this would be a person not carrying the mutation. And then we've got the husband and wife here. So the geneticist then, from this research, told the couple they were both carriers of the mutated disease. Explain how you reached this conclusion. Well, from this conclusion, looking at this, we could decide straight away that the carriers are heterozygous. So they have one normal copy and one mutant copy. So they don't have the condition. So how could they both be carriers? Well, they are both heterozygous. Both carriers are the carriers are heterozygous, which means they've got one normal and one mutated gene. They don't have the condition. That will get you 1 mark. You're explaining basically there what a carrier is. We're going to now explain how we know they're heterozygous. Well, we know they're heterozygous because we know that actually they must have the gene, the normal genes that the person not carrying the mutation has, because some of the DNA probe has bound and has lit up. So we know that straight away. So both have DNA that binds, but that DNA that binds is about half. So both have DNA that binds, have about 50% DNA that binds to the probe. That would be your 2nd mark. To get your 3rd mark, which this question is 3 marks, we then need to explain the probe. Well, what did the question say about the probe? We're going to use a DNA probe to look for deletion mutation. The DNA probe was specific to a particular base sequence in an exon in the gene. Clearly, the DNA probe binds to the dominant gene, dominant allele, because this mutation is deletion and it causes genetic disorder, which is homozygous. So it's going to be homozygous recessive. So our probe is going to bind to the dominant allele. So only 1 copy of the exon is in their DNA, so they only have 1 copy. So each parent only has 1 copy. So that is the first part of that question. Then goes on to ask, and that would get you 3 marks. The DNA probe the geneticist used was for an exon in the DNA, not an intron. Explain why. Well, exons are what code for DNA. Introns are not translated. So that's the first thing we're going to state. And this question, I'm just going to check how many marks this question is. This question is 3 marks again. So introns are not translated. So because introns are not translated, they're not going to be present in mRNA. So they're not present in mRNA. It's supposed to be a little m. So we wouldn't be able to test for that anyway, because when we're doing these DNA probes, it needs to be from an intron, because we get the mRNA and then we turn it into complementary DNA, the cDNA, and we can't do that. We can only do that from the mRNA, so they wouldn't be present to start off with. And actually, these mutations affect amino acid sequences, so they're not going to be present in introns. So all our mutations are occurring. These mutations affect amino acid sequence. This deletion is going to affect the amino acid sequence. And it's going to then go on to produce a faulty protein. That protein is going to be faulty because we've had a deletion, mutation occur. So that whole is going to be a shift change, and everything frame shift change, everything after that deletion has changed. All those amino acids afterwards would change. So because of that, it's important to know if the parents' exons are affected rather than any other part of their DNA. So it's important to know if the parents' exons are impacted rather than any other part of their DNA, as they are what produce proteins. Basically, the point of the question is, why are we looking at exons, not introns? Well, introns aren't translated, not present in mRNA. The mutation affects amino acid sequence, and exons are what produce the proteins. If there's a issue with the amino acid sequence, it would then produce faulty protein. That is then what causes the diseases. So it's important to know actually if those parents have those the issues in their exons, if they're impacted, and so that's what produces the protein. So it doesn't really matter what's happening in the parents' introns because they're not going to affect the proteins that are being made. So that's where the line sort of falls really with that question. And then finally, to make the DNA probe, the geneticist had to find the base sequence of the normal gene. Once he had copies of the gene, what methods would he use to find the base sequence of a gene? There's a couple that we've talked about. We've looked at restriction mapping. If you're not sure what restriction mapping is, have a look back at a couple of the videos. Or we could use base sequencing of fragments. If we did base sequencing of fragments, you would need to name some methods of that as well. But I'm going to leave that one as one for you to have a little bit more of a look at and have a go at answering yourself. I've given you the basic answer to that, but you would need to bulk that out a bit more. So that's one I would suggest actually go and have a little look at. So make sure you know what restriction mapping is. Restriction mapping is the answer, but you need to actually say what it is to get that next part of the mark. Same with base sequencing, DNA sequencing, base sequencing of fragments. Give us a description, give us a method. And that's where I'm going to leave that, just with a little bit of work for you to go back and have a look and make sure you definitely know what those words mean and how to do that.